I have the pleasure to introduce you Jérôme Lecoq. It's a double pleasure. First, because I'm very eager to know what he will tell us about, but also because I'm given the privilege to introduce a talk on observatories, which is a field that brings me into science in the first place, astronomy. So, see you.
Thank you very much for all the organizers for inviting me over. It's a great pleasure to come back to Paris every time, and so I'm grateful to present our work in the last few years. So everything I'm going to present today is on this slide, all the concepts, and so I'm going to briefly go through it and then and show why and how we've built it. So we inspired with astronomy. I think the way the astronomy field has evolved is something that we could aspire to in neuroscience. So that was the inspiration for this figure. We wanted to build an observatory where everybody could come in, and that will create a marketplace of ideas. So that's where you have all of these people talking and exchanging ideas. And then people from all over the world could access the data and through a computer and getting to access the data and validate hypotheses and check their results against ours so that we can all discuss. But also experimentalists across the world could sort of explore with their own instruments a little bit of the brain and figure out that something might happen here, and they might want to come to the observatory to double check their findings. And so I think this is an opportunity to create a place, a marketplace of ideas where we collectively figure out how the brain works. And so the rest of the talk is just to show what we did to hopefully make this happen. If this work. All right. So the rationale, why doing observatories in the first place? So obviously, as neuroscientists, our goal is to understand how sensory information is represented and processed. At the Allen Institute, we're especially interested in the cortex, but obviously it's a whole brain challenge. And in particular, we're interested in understanding all the different layers of the cortex work together. To do this, typically in system neuroscience, you have to design a task. So in this example, it's a simple go-no-go task with a grading discrimination. But if you want to do this, you have to essentially accumulate data about the different components. And at the Allen Institute in the last 10 years, we've accumulated a lot of data about anatomy. We've built all the atlases that helps, I think, everyone to understand where they're recording from. We also started to accumulate data about connectivity within that sort of atlases framework. And over the years, we had a couple of teams that were dedicated to understand the components of the brain, the cells, their trophology, their morphology, and how they are connected to one another, as well as what are the different cell types, how many cell types are there in the cortex. But we were lacking a key core component is how the different cell types act in vivo. That's how we started this project. And before I get into the details, is why I'd like to talk a little bit about why an observatory makes sense in neuroscience. So the first hypothesis is, and I think everybody will probably validate that hypothesis, that we'll likely need a large number of experiments if we want to figure out what's going on, especially if you want to build complex models and train the data on it. The second hypothesis is and I think it's a recurrent problem in the field that talks about reproducibility and all of that. It's a large number of its experiments are duplicated across labs, and so it would be really ideal to have a common reference framework to integrate them, and everything speaks to the end of the sentence. And the last hypothesis is we think that an observatory model will synergize the three main communities: the experimental neuroscientist, the technological, and the theoretical neuroscientist. And we think that because this is what happened in astronomy. This figure here is taken from the Keck Observatory in Hawaii, where there's a pair of telescopes that are watching the sky every night. And this is already kind of old statistics, but if you look at the number of papers that this instrument generates, it's already in a couple hundreds per year. And so the entire community has access, and every night they run experiments for everyone across the planet. And so we were inspired by this, and the rest of the talk is just to show what we've done to try to make this a reality. So we had to put a lot of effort in building a very standardized observatory. And we were sort of really concerned about the issues of reproducibility and also the standardization. And so we had to do a lot of effort towards this. So obviously our goal is to understand sensory processing, but to start with this we had to make a couple of simplifications because we wanted to do this at scale. So that would be hundreds and hundreds of mice. So the first thing we simplify is made the problem a lot simpler with having only animals passively viewing a screen. But let's simplify the training part of it. But I'm going to come back to this at the end of the talk. We built this pipeline but in this way. So this sort of a schematic of how it's processed today. So you start with transgenic animals, and then they're implanted. So every system neuroscience experiment in the field has all of its components, more or less. So we're not different in that regard. So you start with an animal model, you implant the animals, and every inch of this step provide data. But you need to register and standardize. The animal is mapped, so you map the visual cortex, and then you need to habituate the animal so that it is not a stress on the instruments. And this is a step that is done to do neural recording. Now we're using electrophysiology as well. And then there are controlled experiments that you always need to do to check that the animal is stable, that the brain was not altered, and to reconstruct in 3D to reference to our atlases that we have. And I'm not going to speak too much about the details of the pipeline, except I'll show you, to make it grounded, an example data set. So this is an experiment that is done every day. As we speak, people in Seattle are still collecting data on this pipeline. And it runs 8 hours a day. And so on this experiment, the mouse is watching a screen, and you record the neuronal activity that's processing the visual information. And we also follow high tracking as well as a lot of metadata. And I have a couple of pictures of how it looks like. So essentially, it's like normal Twophoton instruments, but just scaled up. And I mean, there's no magic to it. We just build many more rigs, and we have people dedicated to each individual steps. There's a team of surgeons that every day, most of the day, spend doing surgeries. And sometimes they're cross-trained, so you have people that can do surgery as well as Twophoton imaging. But it's really essentially a pipeline. It's a data collection pipeline. Key components that enable a lot of the standardization, I think it's a solution to this problem, is to have a team of dedicated engineers. We have amazing engineer at the island that did all the CAD works. Every step of the process was modeled, and we built a coordinate system that allowed to register the mouse between each individual rigs, so that when the ISI give you a map, you can take the animal and bring it to the Twophoton rig and click on the screen, and the microscope will move exactly where you ask it to go at the center of the area. So, and there's a lot of engineering, and unfortunately I don't have the time to show you all of that, but I thought a little bit how I could showcase standardization and reproducibility in this pipeline. This is a figure that we made recently. So first, you see that to generate some of the data that we recently published, we had to go through hundreds of mice, so that show for the reproducibility of the process and the quality. Every animal is going to very standardized data curation, and we have documents to support it. There are no 350 pages of standard operating procedures that are documented on the pipeline. So there's many documents that explain how you do Twophoton imaging at the Allen Institute using the instruments that we use. And they follow, most of the time, very objective criteria to define when the data is not valid and why it's valid, with a cutoff threshold. So for instance, we have a 10 micron threshold on Z-drift, or we have a criteria to detect epilepsy, and if the animal shows any occurrence of epilepsy, is discarded from the pipeline. We don't compromise. All right, so I hope I've shown you that we've built, I think, some solution. There are other ways, but I think we've provided some solution to standardization and reproducibility and documentation of a system neuroscience experiments. And in the next few slides, I talk a little bit about some of the scientific outcome that we have seen with this data collection effort. So we designed an experiment that our goal initially was to understand how the visual information is processed across the visual cortex. And we designed a set of stimuli that allow us to characterize the visual properties of the cells. And it was made of like natural stimuli, like pictures and movies, as well as synthetic gratings and locally sparse noise. We had to break it down into subsessions, and we went back to the same cells across the 3 sessions. This is the outcome of the data collection effort that we had done in the last few years in numbers of cells. As you can see, we've been able to cover very broadly from the layer 2-3 to the layer 6, many different cell types, including inhibitory and excitatory cells across 6 brain area. And importantly, all the data that we have collected, we release it on schedule with tight data release schedule on these dates. And then I'll talk a little bit about some of the analysis later. But the data was released actually prior to the paper, and you can take your cell phone now and go to the website and access a database of cell responses. And I'm going to talk tomorrow in the workshop on how to use it. But these are characterization of in vivo responses to the visual stimuli. And we didn't talk too much about science today, but this might be a little window. One thing that we noticed in the data that was surprising, and I think it showcased the value of unbiased surveys, as the one we did. And I apologize for the college, they're a little faint. But if you look carefully, these are responses of neurons to different stimuli. It's very obvious when a stimulus is on, so some cells respond specifically to that stimulus. But it's also very hard to predict the response of a cell to this stimulus by looking at the other stimulus. And we are actually kind of surprised by this. And this is the statistics of this effect. So if you look at the response of cells to natural movies, there is very little, you have very poor prediction of the response to the other kinds of stimuli. And we think this is an emergence of how the visual cortex works in the mouse. It's properties that was surprising because if you take a standard models of how the visual cortex works, you should be able to predict if it's only Gabel filters. You should be able to predict by looking at how they respond to gratings, how they're going to respond to natural scenes. And the value in having this data really standardized is you can compare to modeling, and people can start to explore. And Michael Bias and other people in his team started to compare the responses of these cells to a deep neural network that would process the same image. And so in the deep neural network like VGG16, you expect much more and more complicated features as you dive deeper in the network. And surprisingly, if you actually look at the responses of the cells to different blocks of stimuli, you actually see the emergence of this independence of the responses between different blocks, which seems to indicate that the response we see in the visual cortex here are made by a very high order system, as if the visual cortex is already further down in the hierarchy as opposed to just a simple Gabel filters. There's a lot more in our bioarchive paper. I invite you to take a look. Obviously, it's a little hard to, in the 20 minutes, to explain all of that. But you can also regenerate the analysis yourself. So we built a set of Python libraries, the same way as you advise. You can download on a computer and download the data through this library and regenerate analysis using Python code. And the library provides most of the functions that we use to do the analysis ourselves. So this is also a solution to the issue of the reproducibility. One thing that I wanted to showcase is we released this data 3 years ago. And there's already many groups across the world that have been using this for very different questions. And so I think it's a success of open science. Like some people are using it to develop new algorithms. Other people are looking at properties of plasticity by looking at the responses of cells for the beginning and the end of the session and comparing with their coupling with the network. It's very, very varied studies, but leveraging this data in so many different ways. So it's inspiring. All right, so I've shown you that I think we've made effort in building this observatory. We standardized it. We made it reproducible. And I'd like to talk a little bit about the future moving forward with this observatory. The first thing, obviously, we started with a simplification where we didn't have a very strong behavior components, and we put a lot of effort lately to include behavior in the pipeline. And we decided to go with a visual discrimination task where the mouse is detecting a change on the screen. So it could be gradings as well as images. And this is just to showcase that it's not working. And people are collecting data at home with behavior tasks every day now. The other component I'm really excited about is the Twophoton imaging that we use. And I think observatories in general across the world should match the state of the art. And we should provide cutting edge technology to the field. And we've done this in multiple ways. So this is data that we acquired now at the Allen, where we are able to record 8 planes from 2 brain areas of any pairs of area of our choosing simultaneously. And it allows to really look at cortical computation between 2 pairs of areas. And we're gonna actually do this behavior recording using these instruments. And I can talk about the technical details if you're interested. Just ask me questions. All right. Another avenue where we are going to extend this observatory is to electrophysiology. So Josh Siegel here and some other people, he's also involved in the Open Neuroscience initiative very heavily. We designed a system that includes six neuropixel probes and allow to record both cortical area and subcortical area simultaneously, and this data will be released, I believe, next year, as in the exact same condition as the 2P data. All right, but I think there's another avenue where we actually could have an impact on the field is by really making this a true observatory. Last year, we actually piloted this. We opened it up to a subset of scientists. We wanted to know what was the interest and if there was value in actually enabling anyone to come here and propose experiment. This is how we see this process today. It might change, but this is how we see it. So we will have release of RFP, and the hope here is really to create a marketplace of ideas. So incentivize the best ideas in the field that leverage these technical capabilities, and select the best and most impactful science. We made this process blinded to actually remove any bias that are not scientific. So the reviewers that were part of this process were not aware of the identity of the people that proposed their science. I think it's a key component of observatories moving forward. We actually see this as a 2-year process. So the first year, most of the year is actually for writing proposal. And we made a letter of intent step followed by a project, a full proposal review. And after 6 months of work on your side, there's a project planning with the Institute. And then we start to do a pilot data collection, which actually we did last year. So we received proposal in January last year. And we selected the first one in February. We selected for proposal. I'll give you details on this in the next slide. And we went on to do a couple of pilot data collection to help them sort of fine-tune the experimental design, also based on the review. The review process has the advantage of helping you sort of formalize your science better, because in the same way as if you go to a journal and ask them if they will publish this without the data, here they were reviewed by scientists, their peers, before we started experiments. So it's an opportunity to improve your experimental design slightly. And then we collected very briefly few pilot data experiments to validate their experiments, and went on to do main data collection. And we are in this phase of this pilot. These are the 3 projects that we selected. And we are actually right now on the second call. We've received 33 projects so far from a small pool, so we are really excited, and most of them are really high quality. Interestingly, they come from very different backgrounds. So the first project came from a group of really good machine learning scientists with a strong neuroscience background. And they were interested in understanding the credit assignment problem, which is it's a problem that machine learning has a solution to, but we don't understand how the brain does it. The second project was interested in understanding more experimentally what are the stimuli that the mouse cares about. I don't think we know that very much, as opposed to cats or humans. And the third project was interested in predictive coding, which is a very hot model currently, and wanted to validate some hypotheses there. And I don't have the time to show you all of this project, but just to mention that we started, the advantage of having an observatory is you can really streamline the process. So these guys, we started collecting data in May last year, so like a couple months after we proposed the experiment, and we're already done. So they already have the data, they've already done the analysis, and I think they plan to publish this year. Hopefully, depends on their progress. And this group here, also, they're already working data. They had a SFN poster, and some of you might have seen it. So I think it's a showcase of the success of the model. And this group here, we're still collecting a little bit of data. Here, on the next slide, I have a little bit of science. This is from the credit assignment project that we run on the OpenScope platform, the observatory. They were interested in comparing dendrites and somas in response to a sequence of images like A, B, C, D, and then suddenly you change D on the mouse, and it has never seen it before. So it's a very strong way to evoke some form of learning during the session, and they show that they actually can see that phenomenon using the data we collected from them. So there they look at the response of the cells from the early and the end of the session, and show that very strong surprise component, both in the layer 2, 3 and layer 5. And here they're comparing with doing the same experiment with a model that they're running, a simulation they're running on their computer. So to summarize, I hope I've convinced you that we've proposed some solution to the problem of standardization and reproducibility. And also, I think it's an exciting opportunity for the field to come together and create a marketplace of ideas that leverage some of the best tools to answer the most important question in neuroscience. Thank you for your attention. I do have to say a few words. So Paul passed away recently. We're really sad about it. And we're doing our very best to continue his legacy. So I don't think it's going to affect our work too much. But we really regret our loss. And there are a lot of people that were involved in this work. I tried to put them underneath. It's truly teamwork that was involved into this. Thank you.